Hello, everyone, and welcome to today's live broadcast, Better Care with Better Knowledge, presented by Rupert, Associate Director, Genetic Disease Informatics at QIAGEN. I'm Alexis Cross of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, please visit www.qiagen.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Yip. I will now turn the presentation over to him.
Thanks, Alexis, and thanks, LabRoots, for allowing me this opportunity to talk about some of the exciting things here that are happening at QIAGEN Bioinformatics. What a lot of folks don't realize is that QIAGEN actually has a thriving bioinformatics business, but our business is really about the offering of knowledge as opposed to purely bioinformatics algorithm. And so this is why I talk about the theme of our product as better care with better knowledge. So the first thing I want to talk about is this clinical blind spot. As more patient samples get sequenced around the world, we're starting to uncover more novel variants from various patients and patient populations. And in this bubble, I try and depict the universe of possible human mutations as the universe of all possible human mutations. Of course, we're just starting on that endeavor to uncover all these mutations. And you've got your scientists that has their own domain of knowledge, which is fairly limited compared to what's out there. But then you've got these crowdsourced efforts such as ClinVar that try to aggregate this information. As well, there is more organized efforts such as HGMD that's been around for quite a while that has worked to curate all that knowledge from published literature. And then finally, the QIAGEN knowledgebase really aggregates information from public sources such as ClinVar, Omen, and other public data sources, as well as private data sources such as HGMD. And so to my knowledge, the QIAGEN knowledgebase really represents the most inclusive of all knowledge sources out there to bring you as much information and to be as comprehensive as possible on all possible known human mutations. To that end, I want to just throw out some numbers. QIAGEN had acquired several companies, including Ingenuity, BioBase, CLC, and more recently, Omicsoft. And in aggregate, these organizations have now over 13 million knowledge base findings. We aggregate data from over 30 different data sources across 20,000 disease classes. And our knowledge base and our applications have been used in over 30,000 publications. Additionally, our platform has now been used to analyze over 750,000 human samples, which to my knowledge is the most largest experience of looking at human samples from a genetic standpoint. Where do we get a lot of this information? This slide really talks about some of the data sources that we get our information from. Some are public sources that are in the white bubbles here. Some are proprietary sources such as Innova, which is a healthcare system that have made some of their clinical cases available to us. But some of them is our own internal manual curation efforts where we read scientific and clinical papers to pull out things like clinical case counts. And then also from sequencing labs such as CentoMD that have made their cases available through a database called CentoMD. And so, these public and private data sources really represent sort of our effort in aggregating the world's knowledge into the knowledge base. Now, when we talk about knowledge, clinical knowledge, we typically talk about clinical breadth and clinical depth. Clinical breadth can be defined as how large is the domain of knowledge, and that can be measured by, for example, the number of unique genes that are curated or the number of variants that are curated. Clinical depth, however, is really talking about how rich or deep is the knowledge that we're curating. And one way to measure that is the number of citations associated with each gene or variant. And then within each citation, within each paper, what richness of information are you extracting from those scientific articles? Things like clinical case counts, how many patients were affected, or how many were unaffected. If there was drug information, what was the drug? What was the dose used? How many of those patients were respondent to the drug and how many weren't? So all that useful information is manually curated and entered into our knowledge base. So when we take a look at the breadth of coverage, currently our knowledge base has knowledge on variants across over 18,000 genes in the human genome. That represents close to 90% of the human genome. And if you take a look to see where some of that information is from, you can see that some of those gene information we get from ClinVar, some we get from ACMG, but there's a large percent of that information that is through our own organic curation effort. And so folks who are just using ClinVar alone are missing out on more than half of the variants that are, more than half of the genes that are known to the QIAGEN knowledge base, but also to ACMG. Further, if we take a look at the number of variants that are captured in the knowledge base, we have annotations across almost 1 million variants. And as you can see, more than half of those are exclusive to the knowledge base. So again, if folks are just using ClinVar, they are missing out on more than 50% of the information they could be having access through our knowledge base. As an example of how useful this information is, we took a hypothetical panel of 59 genes, the ACMG 59, and compared the total number of variants that you would get if you were to just rely on, for example, the ClinVar database or just the ACMG. Compared to the knowledge base, the ClinVar, the knowledge base has more than twice the number of variants covered in the knowledge base compared to ClinVar. And even with ACMD, folks are still missing out on maybe 20%, 27%. When we take a look at the number of publications, ClinVar doesn't really have publication information. But even with you're just relying on ACMD, you're missing out on 4 times the amount of information because the knowledge base really strives to be as comprehensive as possible and to try and uncover almost every publication for those particular genes that are ever published in the scientific literature. And as a result, we have many more publications for those particular genes and variants that are found in those 59 genes. So what is the importance of having access to comprehensive and timely information? I want to talk about a fairly famous case in 2007 where a clinical testing lab had tested a child's SCN1A mutation, and at the time they classified it as a variant of unknown significance. And because of that classification, the physicians really didn't alter the child's treatment, and consequently the child passed away a year later. Well, the mom wanted to follow up and get some more information, and when she pulled that report, she realized that for that particular mutation, that the lab at the time should have noticed or should have had access to 2 publications at the time of that test. And she claims that back in June 2006 and March of 2007, there were 2 papers that provided sufficient evidence that might have changed the classification of her child's mutation. Her claim is that this evidence, and in fact there was a March 2007 paper that had an exact patient with the same mutation and the same phenotype that her son had. And her claim was that had the lab had access to this knowledge, they probably would have classified that mutation as pathogenic or likely pathogenic. And if that had happened, then the physicians might have altered the course of the treatment for her son, and her son might still be alive. And so that case is undergoing litigation right now, but it's just an example of this blind spot where the labs didn't get access to as much information as they could in a timely manner. So really the take-home message is that whenever you're doing any clinical testing, you really want access to the most comprehensive and timely knowledge in order to do a proper assessment. Now, what can you do with this additional knowledge? Well, one of the challenges with clinical testing is identifying these novel variants and trying to understand the significance of those variants. When there's not enough evidence, clinicians classify these variants as a variant of unknown significance. And these are really challenging because doctors really don't know what to do with these types of variants. It's equivalent to saying that, I'm not exactly sure whether this is a good or a bad thing. And so any effort to reduce the number of VUSs is extremely valuable. We did a benchmark study looking across 180 variants for Lynch syndrome. And compared to how ClinVar classified these variants, our application, our knowledge base, was able to look at these 180 and really clarify the VUSs to the point where we were able to reduce the number of VUS by 20% and reclassify those VUSs to either pathogenic, likely pathogenic, or likely benign or benign. And so this is just a benchmarking example where more knowledge brings greater clarity. I also want to talk about our CentoMD offering. CentoMD is the newest member of the bioinformatics knowledge family. It really is a collection of rare variants based on clinically diagnosed patients from the CentoGene testing lab in Germany. The unique thing about all the variants within CentoMD is that every variant is based on an actual patient. Additionally, the variants are classified by a CAP-CLIA-trained lab director and are based on following the ACMG guidelines. And more importantly, many of the variants in this database is not published anywhere, so you would not find them in, for example, HGMD. So with that, I want to just sort of highlight the product offering for the QIAGEN hereditary disease portfolio. It includes HGMD, Ingenuity Variant Analysis, QIAGEN Clinical Insights, and CentoMD. HGMD is really primarily a survey of all published literature. Ingenuity Variant Analysis is really a powerful tool to rapidly identify causal mutations. Clinical Insights is our clinical decision support tool, and CentoMD is really the world's largest database of rare disease mutations. And I'll walk through each one individually. So HGMD really is strives to be a comprehensive resource of published germline disease-causing mutations. It strives to be 3 things. It strives to be comprehensive in that it tries to record all disease-associated germline mutations that it's in the published literature. It strives to be high quality in that it's created through manual curation. There's a team of highly trained genetic experts who read the scientific literature and extract this information. And it strives to be current because we update the database on a quarterly basis. Now, as more sequencing starts happening around the world, what we're starting to see is an explosion in the number of uncovered variants. And so the ability to keep up with this information is really quite challenging. Imagine a single lab director, even if they have a very specific domain that they're interested in. It's, over the course of several years, it becomes really challenging to be able to keep up with all the published new mutations that are coming out. And so our team is really designed to scale and to capture that knowledge where folks individually may not be able to. And this chart really takes a look at the progression of knowledge that is accumulating. The number of germline mutations published has really doubled in the last 10 years, but we expect the next doubling to occur in 5 years or less. The great thing is that HGMD is available as a web service. There's no software installation that's needed. It's available as a subscription service, and you can just launch a browser to be able to get access to this knowledge. And when you do a search, you come up with a list of mutations, and when you drill into an individual mutation, you get information such as mutation ID, the class of the mutation, a list of the scientific references, and a link out to the publication so that you can actually read the scientific article, details of the mutation, as well as predicted effects based on in silico predictors, as well as orthogonal mutations that are in other organisms. How do people use HGMD? Well, typically there's 2 ways they approach using HGMD. Number one is if they know the disease or phenotype, but want to understand what genes or mutations are affected. A perfect use case is if you're designing a panel of test panel for cystic fibrosis, for example, and you want to understand what genes are associated and what hotspots you might want to put in your panel. Or if you're looking for a panel for Lynch syndrome, what genes you might want to include in that panel. And so HGMD could be a great tool for you to identify a list of genes and mutations that you want to include in that panel. The other way is where you already know the gene or mutation but want to understand what phenotype or disease is associated. So if you're looking at undiagnosed diseases, hereditary diseases, and want to understand what phenotypes or diseases associated, you can use HGMD to quickly identify those phenotypes of diseases. Really, HGMD is designed to help users save time by quickly identifying the disease, mutations, or the disease and phenotype associated with those mutations. Here's an example use case where if you're looking up all the mutations for cystic fibrosis, one way you could do it is to go to PubMed, type in the term cystic fibrosis, and that will return around 3,415 articles. And if you're able to read really quickly, on the order of 5 minutes per article, that still takes you 284 hours. And at the end of that exercise, you'll come out with a list of 1,383 mutations. Alternatively, if you use HGMD, you could type in the term cystic fibrosis through the web browser, and within a 5-minute search window, return the same number of 1,383 mutations. So just to summarize, some of the key benefits of HGMD is really the assurance that the mutation found in the HGMD database is disease-causing. The fact that that mutation is found in the database means that it's disease-causing. And so the benefit of that is that it could take you a really long time to get that information. Just let us do that scouring of the literature for you. Again, HGMD can save you hours or days of surveying the literature for a particular gene or disease. And using HGMD, you could really identify the disease-causing mutations within a gene, or the mutations associated with a gene, or the mutations associated with a particular disease within minutes. And while HGMD is a paid-for subscription service, you do save money from the amount of time that that you would have spent surveying the literature, but also that you don't need to subscribe to every journal article. We can just find the article that you need, and then you can just go and purchase that one article as opposed to just the entire subscription. Next, I want to talk about ingenuity variant analysis, which is really a tool to rapidly identify the root cause of specific disease or phenotype. Now that folks have started sequencing whole exomes and genomes because the cost of such efforts are much reduced, we're starting to see a lot of folks that are using whole exome and genome to try and diagnose rare diseases, undiagnosed diseases, looking across not just the affected individual, but maybe in a trio where you're looking at the affected individual and their parents. Oftentimes when you're sequencing a genome, you're looking at the order of millions, 4 to 5 million variants. And it's really hard to know which set of variants are responsible for the phenotype or the symptoms that is presented in the patient. With a tool like ingenuity variant analysis, you can quickly filter down from literally millions of variants down to the 10 that are likely causing the disease. And we do this through a set of steps where you can reduce variance through common sense questions like, show me only variants that are not present in the general population, because presumably if they're present in the general population, it's not causing disease. And that's what the common variance filter does. Then you might want to say, well, show me variants that have no obvious effect on protein, because if it doesn't affect the protein, it probably isn't going to impact disease or cause disease. Those tools up there are probably not unique to IVA, and a competent bioinformatics team can probably reproduce a pipeline that does that. The lower portion, though, is really making use of the knowledge base, where you can identify relationships between diseases, pathways, biomarkers, and so forth. And so these filters down here really allow you to granularly filter down to the few variants that are likely to cause disease. And I'll walk you through some of those filter settings. So again, variant analysis, IVA, is available as a web service, so no software is needed to be installed. And on the left here, you can see a cascade, what we call a cascade of filters that allow you to, at each step, rule out certain variants and include variants that are interesting for follow-up. So the first step is just identifying a confidence filter, and a confidence filter is essentially saying, I only want to look at variants that I have high confidence in, meaning that they have a good quality score, enough read depth, and are not in a noisy part of the genome where I can't really trust the data. The second step, the second filter really is asking that common variants filter, where you're saying I only want to look at rare variants that are likely to be present in an affected individual and not in the general population. The third filter allows you to ask questions like, show me only those variants that are likely to cause disease, and you can filter by pathogenic or likely pathogenic as defined by ACMG, or filter for those that are present in HCMD or found in ClinVar. So these or look for variants that are likely to cause gain of function or loss of function based on evidence in the literature or based on in silico predictors. So a lot of these tools, a lot of these sort of filtering criteria is something that is not easily doable on a resource that uses public sources. So things like ACMG is probably something that is not so easy to compute, but certainly to look for things that are established in the literature, unless you've manually curated a knowledge base of scientific literature, you would not be able to do that type of filtering. another really cool filter is to say, show me only the mutations that are present in the child but not in the parents. And you can look in terms of homozygosity, heterozygosity, compound heads, and so forth. So this level of flexibility to ask these types of questions is really unique to variant analysis. And then finally, you can then rank those variants based on a list of phenotypes and see how many of those phenotypes are matched with that particular variant. And we are able to do that match because we've read scientific literature and identified various phenotypes that are associated with various mutations in the published literature. And then once you identify that short list of variants, on the right you can click to see more details, and that will quickly bring up the supporting literature for those particular variants, essentially the annotations of that variant, including the published literature that matches with that variant. And you can just click on those links to go and read those papers and look at the evidence yourself to see whether you believe that particular mutation is relevant for the disease or phenotype that you're studying. So again, the key benefits of variant analysis really is that it allows the biologists to do this type of querying and hypothesis testing on their own without the need for scripting or programming. Researchers really can control the filtering settings and do iterative hypothesis testing through an intuitive graphical user interface. It puts the variants and genes into biological context such that you can ask that question, given this list of variants in my short list and the list of phenotypes that I see, show me only those variants now that have any relationship to the phenotype that I'm looking at. And so that sort of filtering based on phenotype and biological context is unique to variant analysis. And finally, because variant analysis taps into the knowledge base, it really gives you access to the world's most comprehensive knowledge base to answer these questions. But don't take our word for it. We have a lot of researchers around the world who have been using variant analysis now, and they swear that this tool provides valuable resources to quickly identify and really save time. But also, in some cases, this knowledge base allows them to connect the dots and create new hypotheses from their data. Next, I want to talk about our clinical decision support platform called QIAGEN Clinical Insights. It's a tool that's used with at least several use cases, rare and hereditary diseases, hereditary cancers, and somatic oncology. The unique thing about QIAGEN Clinical Insights is that it incorporates not just the ACMG classification scheme, but also the AMP-ASCO-CAP guidelines for tiering for somatic cancers. And it uses a computed classification and tiering scheme in order to automate the classification and tiering of hereditary and somatic cancer cases. It, again, is a web-based application with a graphical user interface that makes it really easy to quickly identify the variants of interest to generate patient reports and to make clinical decisions on. It provides instant access to the evidence. So for example, this particular variant was classified or computed to be pathogenic, and it provides evidence such as the effect on the protein, any reported clinical cases that have been seen before, either in the lab or in other laboratories. So in this particular example, it said that this particular variant has been identified in ClinVar, in COSMIC, and in HGMD, as well as information on rarity in the general population and in silico predictions underneath. To understand how that classification of pathogenicity came about, you can click to look at the criteria that are triggered. And so in this particular case, this variant had triggered PVS1, PM2, and PP3 criteria. And you can click to look at the scientific literature to understand where those criteria come from. In somatic workflows, you can actually see the number of patients that have been reported with that same mutation. And if you click on those particular mutations, on those particular patient icons, you can actually get the list of publications that refer to those particular patient cohorts, and then you can drill further in to identify, to read about those particular studies. Once you've done your due diligence of looking across the literature and the clinical cases, you can then review some of the scientific literature to see whether you believe the criteria that are triggered and the supporting evidence. And when all that information, when you've done that due diligence, you can then begin to sign out, or you could, in this case, look at some of the literature and say, you know, I really believe this case, believe this publication, or you could say that you don't believe this publication. I might want to exclude this from consideration in future cases. And so all of that information can be captured to really streamline the workflow for the next time you encounter this particular mutation. Once you've done all that work, you're ready to sign out. And to do that, you can just quickly select that particular case and do a sign out and print a PDF of that report, and we can customize that PDF report with your particular lab's logo and the information that are created so that you can deliver the report to the requesting physician. So QCI, QIAGEN Clinical Insight, really takes advantage of the knowledge base to look across as many genes as possible, and because we have knowledge of really close to over 23,000 genes, it really gives you the assurance that you have access to the most comprehensive resources if you're looking at whole exomes or genomes. Because we've got knowledge of and experience of having seen over 700,000 patients, you can benefit from the experience of having seen virtually 700,000 patients by virtue of tapping into our knowledge base. Something else I want to highlight is that because of our manual curation and our experience of using knowledge from the CentoMD database as well as from other labs, we can actually trigger the PS4 and the BS4 criteria. And this is important because while many platforms out there claim they do ACMG assessment, very few actually trigger the PS4 and the BS4 criteria. And this criteria really is to say that, you know, there has actually been clinical cases that have this exact mutation and this exact phenotype. And when you have real-world examples, that's when you trigger the PS4 and the BS4 in the ACMG classification. And the QIAGEN knowledge base brings that evidence so that those criteria do get triggered. Our manual curation effort really means that we can identify patients in the published literature that responds to a particular drug based on genotype, and this really helps provide confidence in the triggering of those AMP and ASCO cap tiering calculations. We also have access to a broad mix of ethnic populations that are unique to the knowledge base, and this is because we have access to certain ethnic cohorts that are not publicly available, and this really gives researchers, especially for those underrepresented ethnicities, a better way to discriminate between benign polymorphic mutations from actual pathogenic mutations. Now, for those of you who already have their own in-house pipeline and reporting system and don't need the full power of QIAGEN Clinical Insights, you can actually access the knowledge base through our API. And so this is another option of getting access to the knowledge base as opposed to buying a full-blown QIAGEN Clinical Insights. And so the way you would do that is use your pipeline to identify a short list of variants, send those variants via API call to our knowledge base, and then we would return that variant with all the knowledge we have on those variants, including our comprehensive computation of ACMG or AMP-ASCO-CAP guidelines. So again, to summarize, really the value of QCI is really to allow your organization to scale and grow your testing volume, and we can do that through the interface or through the flexible API. Because we've got experience of looking at, there's a mistake here, over 700,000 patients, a small lab can really scale up and execute to the same level as a more established large lab. We also make the ACMG and the AMP-ASCO-CAP computations transparent by providing the evidence behind those computations, so it's not a black box. It's one of the hallmarks of our offering that we make everything transparent in how we calculate those classifications. And really ask around how many platforms actually trigger the BS4 and the PS4 classification, because unless they actually have that evidence from prior labs or from the published literature, it's really hard to make use of that criteria in the ACMG classification. And finally, I want to talk about CentoMD, which is our new database. CentoMD really is the world's largest mutation database of rare diseases. It now contains over 6 million unique variants from representing over 1 billion identified alleles. The patient samples have been sequenced based on Sanger methods or NGS methods or PCR methods. There's also some supporting biochemical analysis as well. And more importantly, every patient in that database has been consented to be recontacted. So this is really valuable for pharma or biotech research who identifies a particular genetic marker and may want to follow up with those patients for additional studies or even recruitment in patient trials. As I said, the lab directors classify all these variants based on established ACMG guidelines, based on looking at public sources, but also their repository of unpublished data as well. And when they get updates every quarter, and those updates really can also result in reclassification of past variants, and typically it's around 2% that get reclassified. When we take a look to see at how many of these variants are known in the published literature, we see a very large, significant part of those variants that are not found in the published literature. So in aggregate, CentoMD contains around 58% of variants that are not found anywhere else. But if you look at certain rare diseases, that level of unpublished variants can vary as high as 69% or as low as 41%. So CentoMD is available as a web service. So it's a web page you log into. You can identify variants based on genotype or based on phenotype. To do a genotype search, you just type in the gene of interest, and it will return a list of variants associated with that gene, and you just activate a variant in order to identify details within that variant. So details that you get access to are, for example, how many patients have been seen with that particular variant, the ethnicity of those patients. You can drill into the individual and identify co-occurring mutations with that particular individual. If you want to look at the variant, see how the lab directors at CentoGene classified that and their rationale for classifying that particular mutation. And then also look at other affected individuals that they may have that particular mutation. And in many cases, you'll have information on family members as well. To do a phenotype type of search, you just type in phenotype terms, and we accept HPO terms. And when you type in those terms and do a search, it'll return a list of patients that are associated with those phenotypes. And when you drill down to each individual patient, you'll find that, for example, these are the list of genes that are associated with that particular phenotype. And then the patients that have those particular variants within those particular genes, and then you can click to identify the variants within those particular genes. So here's an example use case. You want to find out information about this particular mutation in the GLA gene. So when you look at public sources, you'll find that using in silico predictors result in inconsistent results. Some predictors will say that it's benign, some predictors will say it's pathogenic. Looking at Nomad, you see that this particular mutation has never been described, so Nomad's not really helpful. Looking at other external databases, you don't see that information as well. So you turn to a resource like CentoMD, and when you type that particular mutation in CentoMD, come back with 3 individuals with clinical suspicion of Fabry disease. CentoMD even has enzymatic data on all 3 and showed that in all 3 enzymatic levels were decreased and biomarkers were elevated. And this is how and then the CentoMD lab director classified this particular mutation as pathogenic, and here's the evidence that they put together. And so a tool like CentoMD, for example, is able to provide a lot of detail that public sources was not able to provide. Another example is this particular mutation in the GBA gene, where a prediction software said it's likely benign, but at least one published source says that it's disease-causing. So what does CentoMD say? When you type that particular mutation in, it identified 109 individuals, and some of those were colocalized in cysts with other known pathogenic variants. So based on those evidence, the lab director at CentoMD classified this as likely neutral. And so it's really nice to be able to get access to a database where you can ask that question, has anyone else out there seen this particular mutation, and if they did, what was the phenotype, and ultimately, how did that lab interpret it? And so that's really the beauty of CentoMD. CentoMD is a very large testing lab in Germany, and they are adding on the order of 20 to 30,000 new patient samples per quarter. So across a 1-year subscription to CentoMD, you could be getting an additional 100,000 patients into the database. And so that 1 year gives you access to all that information that is added on a continuing basis. So the benefits are really an interactive user interface, allows you to search by genotype or by phenotype. You can drill into the individual to get the information. You can take a look to see how the CentoMD lab director classified those particular mutations and provide the rationale. And you get access to not just curated data, but unpublished data as well. And every time there's a mutation that is reclassified, you'll get an email notification to let you know that that mutation has been reclassified. And then an export function for you to download and put into your own reports or into your own pipelines. And so with that, I'll wrap it up here and take any questions that anyone may have. Thank you.
Thank you, Dr. Yip, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, how is your ACMG classification different from other platforms' classification algorithm?
Yeah, well, so ACMG published their guidelines, and so they were very clear about how you go about interpreting that. And we essentially took those rules and made them into algorithms to compute. But those rules are very specific. For example, they say that real-world evidence will always trump predictor or in silico evidence. And so the unique thing with how we do our computation is that we take into account as much real-world evidence as we can, and we get that real-world evidence from manual curation, but also from historical evidence from other labs who have seen this. And examples of historical evidence from other labs is the abundant data from CentoMD, but also from the community of users who use QIAGEN Clinical Insights to look and bring that knowledge to be shared among the community of users. So, our ACMG computation is based on initially the guidelines that are published by the ACMG, but enhanced with the sort of manual curation and in some ways machine learning algorithms that we have that's part of the knowledge base.
Our next question is, what is the source of the content for your knowledge base?
Yeah, so, we get there was a slide that I talked about. It's a combination of public and private sources. So public, obviously, everyone has access to public sources, and it's just a matter of doing the grunt work to roll those sources into your pipeline. But we also have a rich private manual curation effort, and I call these manual curation efforts kind of a moonshot effort because not many organizations can actually spend the time and resources to do that, because you're not just paying people resources, but you're also paying for journal subscriptions. We have on the order of over 200 MDs and PhDs who scour the scientific literature and extract this information, going through over 2,000 articles a month. That information is then sort of put into the knowledge base. So we get information from both private sources as well as private manual curated efforts that we do on our own.
And Dr. Yip, it looks like we have time for one more question. Why is manual curation so important compared with the machine learning and natural language processing?
Yeah, so we get a lot of there's a lot of news these days about machine learning and deep learning and artificial intelligence, and I just want to give an example where you could get a machine learning or natural language processing algorithm that can read a newspaper article and identify that John F. Kennedy was assassinated in Dallas. And so it can tell that, there was a person named John F. Kennedy and he was killed in Dallas. And that information is then captured with, machine learning or artificial intelligence. But it takes someone with contextual knowledge to know who John F. Kennedy is. So my point is that while machine learning and deep learning can know who was assassinated, but the relevance and the significance of that is something that you require context. And that's why all of our curators are at least MDs or PhD level skill to do that curation. I don't think machine learning and natural language processing has quite gotten to the level of sophistication as a person with an MD or PhD degree. And more recently, an announcement by IBM Watson to sort of scale down their IBM Health efforts really speaks to the sophistication we have today with machine learning and artificial intelligence, and that is that while it's very promising, we're probably not going to replace human curators at least for the next couple of years. And so we want to provide a high-quality product, and that's why we're committed to focusing on manual curation for now until the technology for natural language processing and artificial intelligence improves. That's not to say we won't do it in the future. It's just that for now, we still believe that years, decades of training is still the best way to provide high-quality clinical decision support data.
I would like to once again thank Dr. Yip for his presentation. I would also like to thank QIAGEN for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.